him. Maybe you have many questions about this topic, especially mangrove ecosystem services. So for these sessions, we have about 2 hours for a lecture and a discussion. I give the time to you, Professor Martin. If you can finish 1 hour for presentations, and we still have more time for discussion. So the time is yours, Professor, please.
Thank you very much, Yaya, for the nice introduction and especially for the invitation to talk to you today about ecosystem services of mangroves. When I prepared this presentation, this lecture, at some point I decided well, of course I will talk about ecosystem services, but I will not that much talk about the services themselves, but more about the role that different mangrove species play in there. So the topic, let me check. No, that's not what I wanted. I'm sorry. Here we go. So the topic is more about, as I said, the role of species composition and the ecosystem status or ecosystem health, if we want to stick to the title of this training that you're participating, because in the end, I think this is one of the more important aspects we have to take into account when we want to not only understand, but to sustainably manage ecosystem services of mangroves. I guess you all have quite some background in mangrove ecology and mangroves, but I still just want to bring us all on the same level and want to tell you a bit about mangroves in general. You know that they are restricted to the tropical and subtropical region, very roughly, with a limit at the northern distribution at about 30 degrees north and about 30 degrees south. We see here on the southern west coast of Africa and here on the west coast of South America that there's no mangroves. And the simple reason is that here we have the cold Benguela current and here we have the cold Humboldt current, both preventing mangroves from colonizing these areas. What is important for us today is a very clear biogeographical distinction of the mangrove communities worldwide. We have the Atlantic East Pacific, so the Atlantic space, and the west coast of the Americas, which is the east of the Pacific. And here we have relatively few species, in total a maximum of 15 different mangrove tree species. But even this 15 doesn't mean we have 15 species anywhere we go. In most places, and especially those that I will show you in a second, we have essentially 3, maybe 4 species that are abundant and common. On the opposite, in the Indo-West Pacific, we have clearly more than 60 different mangrove tree species, and we have super high diversity, especially around Indonesia and this area over there, with a very high species diversity of mangrove trees. So the question that we should keep in mind when it comes to the Mangrove Health Index and to ecosystem services is whether this difference in species diversity, species richness between the Atlantic East Pacific and the Indo-West Pacific has any consequences for the ecosystem services the mangroves provide. And before we get into that, I would just like to show you a couple photos to depict more clearly the difference that we find in these area or in the different areas, because it's not only the species richness, it's also the environment that can very strongly differ between the different places that we see. Let's start in Mexico, west coast of North America, close to the northern distribution limit. This is Baja California, the peninsula that is very arid. And I have to say, this is one of the most peculiar mangroves I've ever seen. This is an Avicennia, a forest of 3 species, Avicennia germinans, Rhizophora mangle, and Laguncularia racemosa. The trees are, because of the aridity, relatively small, rarely more than 2 or 3 meters. But what is most peculiar is really the vicinity to the desert vegetation that we see right here. So you have the mangrove on one hand, close to the water, and then with 5 or less meters distance, we suddenly have a desert. We look at the very same species on an island, a Colombian island in the Caribbean. That's the island San Andres. And here again, I have to say, this is one of the most beautiful mangroves I've ever seen. Huge trees, mostly of Rhizophora mangle, but we also have the other two species here. The trees grow easily 30, 35 meters high. And there's a dense network, very complex architecture of roots in this place. It's the very same species, but they look totally different. And the total environment looks completely different. And a couple 100 kilometers further south on the northern coast of Brazil, just about at the mouth of the Amazon River, again, the same species, again, very big trees, very dense forests on a extremely soft sediment, as you can see down here. If you want to work there, you sink in the sediment hip deep very easily. We have to pass channels to work there. And this, of course, you know from other places of the mangroves, but again, same species as I showed you before, still totally different. And the last example from this area is Senegal, again, relatively close to the northern distribution limit on the west coast of Africa now. And here we have very dense, but again, small trees of mangroves, small forests of mangroves, rarely more than 4 or 5 meters high. And on the terrestrial side, we have a very close vicinity to the savanna of the African hinterland. So you might have heard about the Baobab, this very enigmatic trees that grow in Africa and other places in the world. And they're very close to the mangroves here, also characterizing them as a very specific environment, rich, especially in the bird fauna, but also rich in crabs and fish. So that was the examples for the Atlantic East Pacific. Now a couple examples for the Indo-West Pacific. We go to South Africa first. And here we have, first of all, as I mentioned before, different species. We have Avicennia as before, but it's a different species and it grows in a different way. We have these very enchanted forests. We have another species here that's Bruguera gymnorrhiza in the area, growing very tall, sometimes very dense stands. We have salt marshes around and very high crab density in these areas. We stick to the southern distribution limit, but go to Australia near Melbourne. One species, Avicennia marina, growing again, very stunted, very small, rather bushes than trees, close to the southern distribution limit. And just a couple kilometers further north, still Australia, you see, again, very luscious forests, relatively rich in species, and dense vegetation, tall vegetation. Even taller or even denser, I should say, in Fiji, about the same number of species, very similar species composition, very dense root systems here, very dense stands across the shoreline, very high recruitment of young treelings, and you can see the roots here are very high, very dense, but also very high that you can easily walk through. And then finally, Singapore, where we have similar rich flora as in Indonesia. I'm sorry I cannot present any pictures from Indonesia because embarrassingly I've never been to Indonesia before, but this will change. We are starting a project next spring in Indonesia. What I want to show you here is the high diversity in this picture. You can probably easily find just in this picture 5 or 6 different mangrove species, and the whole area, even though very small, is very rich. It is very small. It is threatened by human activity, as you can imagine from this picture, and it's also rich in animal diversity. And this animal diversity is not only, as we can see here in Honduras, vertebrate diversity, but even higher than that is the diversity of invertebrate, mostly crabs that are very rich, both in abundance and in diversity, and very important for ecosystem processes in the mangroves. I will not talk about crabs too much today, although we do quite some work on their ecology. But just keep in mind, they are important players in the mangroves, and they are probably important drivers of some of the ecosystem services. So you've seen that mangroves can look very different depending on where you go and depending also on the perspective that you take. But they all have in common that they provide some ecosystem services, goods and benefits to both local and regional populations and societies, but also to humankind worldwide. You probably know this table, you've seen it before, and it's not exhaustive. We could talk about much or many more different ecosystem services. For instance, when you go down to the social or societal services, recreation and education is just one. It's of course cultural and religious aspects that we have to take into account when we talk about ecosystem services of mangroves in some cases. I will just go through this with a couple examples, these different ecosystem services mentioned here. And I will then get into the topic that I promised to you, that we will talk about more the species that drive that. And again, I will keep that relatively short. We could easily talk about this way more than an hour, but maybe we can just come back to this in the discussion. So habitat for life, for wildlife, I showed you a couple slides with vertebrate and invertebrate species that live in the mangroves before. We saw birds and crocodiles, we saw crabs, there's a lot of snails, different species of snails in the mangrove, so I won't get into that topic too much. Very important is the provisioning of wood to the people living in or next to the mangroves, and wood can be used for timber, for constructing, for instance, houses or areas for landings with the boats and storing the boats for the fishermen. I've seen both in Brazil and in Fiji these fish traps built from mangrove stems that have been used for decades, if not centuries, by the people living there. And of course, firewood. So mangroves are used, the wood of mangroves is used for firewood itself, for cooking, but also for the production of charcoal. And that's, for instance, a very big industry in Malaysia and in other places, as I know, but this really brings quite some income to the people living next to and from the mangroves, even though we have to take into account that this is potentially destructive for the mangroves. So this is an ecosystem service, the use of which, or the overuse of which, very easily and very rapidly leads to decline of mangrove areas, to degradation of mangroves, and even the die-off of the mangroves. This is something we should keep in mind when we talk about this ecosystem service in particular. That's not necessarily true for the provisioning of food. So mangroves are very important as a food source. This is first of all fisheries. Fisheries do not really take place in the mangroves themselves, but just in front of the mangroves, in estuaries and in coastal waters, because the mangroves are very important habitat and nursery for many seafood species, fish, shrimp, crabs, name it. Very different, very diverse marine fauna that spends part of their life, mostly the juvenile part of their life, in the mangroves. And without the mangroves, the abundance of fish in the coastal waters, the abundance on which the people living there depend on, would drastically be diminished. There's also crabs as a very important food source at sea, at least in some areas of the world. This photo was taken in Brazil, and there, these big mangrove crabs are really important as subsistence food for the local population. They also sell them on the market, but they mostly make their own living out of it, their own nutrition. And then in some areas, the seeds of the mangroves are used as vegetables or as seeds. Rhizophyra is one very important example from the South Pacific, but many other species also provide fruit or vegetables that are eaten by local communities. The next point I want to stress here, I want to expand a bit on, is shoreline protection. You all know where and how mangroves grow, but when you look at this picture, just very tiny waves, but still the mangroves growing right in the middle of the water, on the shoreline or in front of the shoreline itself. We can very easily imagine how well that protects the coast from erosion and from damage upon storm surge. As you can see here in this rather extreme picture, the area here on the left where we have mangroves and on the right where we have mangrove and some kind of construction seems to be protected from the strong storm waves, while in the middle where we have this opening in the mangrove stretch, the water body, the waves enter landwards pretty well and pretty wide and have quite some destructive power, as we know from several cases of storm surges or even tsunamis. So even against tsunamis, mangroves might protect. Of course, a tsunami will destroy the mangroves, but we know from the very unfortunate example about, when was it, 16 years ago, that villages, the places that lived, that were situated behind the mangroves, were much better protected from the incoming water masses than areas where the mangroves had been gone, had been degraded, or even cut by people. So this is a very important aspect, and I will come back to that later. And finally, on a more global level, of course, carbon sequestration, or I should say not only carbon, but in general, sequestration of organic matter. Carbon sequestration is important in the context of climate mitigation. It's mostly CO2 that is taken from the atmosphere, extracted from the atmosphere and stored into the biomass of coastal vegetation, mangroves, salt marshes, seagrass beds, macroalgae, anything like that. And then at some point from the vegetation, from the biomass of the vegetation, it enters the sediment where it is stored in a stable form, and I will come back to that later. But it's not only the carbon dioxide, the CO2, that is important here. In the sediment, we also find huge amounts of methane in some places, and we find huge amounts of nitrogen that, if released to the atmosphere as dinitrogen oxide, would also be a very potent greenhouse gas. So it's not only carbon sequestration, even though this blue carbon topic is in the press and media everywhere. So I will, starting from this, I will now give you just a couple examples of places where we did some studies and where I want to talk you through to show you that when we talk about ecosystem services, we have to consider many different aspects. For instance, the status of the ecosystem, and it's quite obvious that a healthy ecosystem is better in providing ecosystem services because ecosystem processes in these systems will run much smoother and will run much more efficiently than in degraded or unhealthy ecosystems. So being able to determining the health status of a mangrove is very essential for understanding the provisioning of ecosystem services. Let's start in northern Brazil with two short examples. I will not provide too much details here. The first one is about the wood provisioning of mangroves. These are freshly cut stems of the species Laguncularia racemosa. And this species is interesting for the people there because, first of all, the wood is very resistant against decay. So this is what people use mostly for their fish cages. And these stems in the fish cage, in the traps, and the fish traps remain for a couple of years, but every now and then they have to be replaced, and the people go into the mangroves and very selectively cut stems of trees that have the right size for what they need to replace stems from their fish traps. And for this, mostly the best suitable species is Laguncularia, because the stems are very straight, and which is a topic that we have been working on for quite a while now. In contrast to many other species, they regrow. So if you cut a mature tree, then after a while, within 1 year, it will start resprouting with several branches, and one of these resprouting branches will then in the end develop into the main branch, the secondary main stem, and the other branches will be gone. And then after about 10 years or so, these trees look almost the same as they were before as mature trees. So it's important here that not all the species in the mangroves are the same when it comes to using for wood, especially not, or at least not in this region. And here it is mostly Laguncularia racemosa, the one species that people need here when it comes to using wood for mangroves. The other example is about the food sources of mangroves, and I told you about crab fisheries, again in Brazil. It's the very same area, the Hydrodynamic Peninsula in northern Brazil, where we worked quite a bit over the last couple of years. And this is a study where we equipped crab fishermen, crab collectors with GPS trackers to see exactly where they go, how they behave in the mangrove, and how much time they spend. And I won't go into any detail here. We believe we've figured out that they act as optimal foragers, a very important term from ecology, describing that you invest more time and more energy in sampling, in collecting in areas where there's a rich catch, meaning many crabs and large crabs. What I want to show you here is again the species specificity of this aspect, because many of the places these fishermen go by boat or canoe are places that are in Rhizophora forests rather than in the other two stands like Avicennia germinans or Laguncularia. And the reason we believe for this is very clearly that, as you can see here from this nice paper from Piu et al., is that in Rhizophora stands, there's not only way more crabs per meter square, so we have about 1/3 of crabs more in Rhizophora than in Laguncularia or Avicennia. And also these crabs are significantly larger than in Laguncularia or Avicennia. So it's more and larger crabs. And this is, we believe, the reason why crab collectors mostly go into the Rhizophora forest, even though moving in a Rhizophora forest and carrying heavy loads of crabs in a bag with you is much more difficult than in Avicennia or Laguncularia stands. So it must be worth this. It must be worth it. And the idea is that for this reason, if you want crabs in that area, the best stand, the species that best provides this ecosystem service would be Rhizophora mangle and not the other two. Which in the end, if I may already head a bit forward to one of the final messages of this presentation, which in the end means that if you want to restore or rehabilitate a mangrove, and the reason for restoration here is the production of food as crabs, then it would be wise to invest in Rhizophora mangle rather than in the other species. Okay, let's talk about shoreline protection for a while. An example from Florida, as you can see here on the map, it's a study that where we have quite dense stands of Avicennia. This picture is a bit misleading because in most of the area, again, remember, as you can see here, we are close to the northern distribution limit. In most stands, the trees are rather small, but they have a dense network of pneumatophores of their aerial roots. And we were lucky enough to be part of a field experiment that you probably can only run in the US and in hardly any other country, because our colleagues there chose areas of mangroves here on the left where they cut different percentages, different proportions of mangroves. So this is a control area where no mangroves were cut at all. And then we have something like 10% cut, 20%, 30%, 40%, 50%, all the way up to 100% cut. And then with this, over years, we followed the development of the fauna, the flora, the sediment characteristics. And the only thing I want to show you here in terms of coastal protection is the observation that compared to the control stand with no mangrove cutting, where we have a little berm at the coastline and high, well, not so high, but as I said, stunted but dense mangrove vegetation. Compared to that in the area where 100% of the mangroves were cut, the berm was removed and the shoreline retreated by about 5 meters within 5 years. So we have a shoreline retreat of 1 meter per year in an area where there's relatively little wave movement. It's a quite protected area. There's a bit of boat traffic, but not very heavily. And still within 5 years, the shoreline retreated by 5 meters upon mangrove cutting, mangrove clear felling. So there is a very clear evidence that mangroves are important for that. And in this case, the effective species here would be Avicennia germinans. I will come back to that later in a second. So what we did in another place in Fiji, we talked to people from a village. There's a little fisherman village or fisher village just behind a very interesting beach or shoreline, because the shoreline is kind of a consolidated, solid sediment. And the mangroves there mostly grow in crevices. So not so much in the soft sediment itself, which is rare at these places, but in these crevices. And the people in this fisher village up here, at some point, started planting mangroves, and you can see here a little plantation in front of the village that is about, or that was back then, about 5 years old, and this one is about 20 years old, in order to protect the village from waves and storms. So that was quite clever, but nowadays they're thinking of building a concrete wall somewhere here in this opening, and we discussed with them that, why it would not be a good idea to do that, but rather invest more in mangrove plantation. And this, you again can see here, the relatively recently planted mangroves. So what we did to convince them is we went to the place, and here you can see a little drone image of the area. This is the about 20 years old patch down here, and we placed sensors or loggers for water height, water inundation level in these areas all around these green dots to follow the wave height in place. What I show you here is a comparison between the side inside the red, the green, bluish one, and the side in front of the mangroves, the orange reddish one. And when you look at wave height, then you see that there is a slight change in wave height between in front and about, what is it, 10, 15 meter inside the mangroves. So we have more higher waves and less small waves outside the mangroves, and the opposite true inside the mangroves, the blue bars here. But the difference is not really big, surprisingly small actually. And when you look at the average wave height overall, then we see there's essentially no difference. The difference is very small between here inside the mangroves and in front of the mangroves. So we were quite disappointed by that, because we expected a much stronger difference, because we know that mangroves provide the service of wave attenuation, or the process of wave attenuation, resulting in the service of coastal protection. But then we took a second look, and we did not look at the individual waves, but we looked at gravity waves. And these gravity waves are groups of waves that travel with the water masses towards land. So it's not individual waves, but group of waves. And sedimentologists, which I'm not, tell me that these gravity waves are the ones that mostly cause erosion. So here we see that within or inside the mangroves, we have gravity waves that are about half as high as outside the mangrove. So when it comes to those waves that cause erosion, just 10, 15 meters of mangroves, in this case, this is a mix of Rhizophora and Bruguiera, cause or make quite a difference. And along the same line, we also see differences between species. This is a study with anymore, but it compares wave attenuation between just two species that were abundant in the area where they did this study. And you can see here the increasing wave attenuation from the sea side of the mangrove to the land side of the mangrove. And you see that these two different species, Soneracia and Candelia, very clearly differ in their efficiency of attenuating the waves. So a 50% attenuation is reached by, say, 200 meters of mangroves in a Candelia stand, in a Soneracia stand, but it takes about 400 meters in a Candelia stand. So again, very clearly differences in species. And if the aim of, for instance, restoration was to increase coastal protection in your area, then it would, in this case, in this area, be advisable to plant Soneracia rather than, for instance, Candelia. And if you remember the slide, or one of the slides before, where I showed you the Avicennia, it's interesting because Avicennia and Soneracia have very similar root structures that obviously are very efficient in slowing down the waves, and by this protecting or stabilizing the sediment. Okay, the last example that I want to talk about in a bit more detail is carbon sequestration as an important climate mitigation factor, not only regional, but this is really an ecosystem service of mangroves that is relevant globally. I mean, the others are too. When I think about seafood in Germany, we benefit a lot from seafood capture and production in many tropical countries, including mangrove countries. But it's more important for the local people. But carbon sequestration, climate change mitigation through blue carbon, of course, is of high relevance and increasingly high relevance for humankind worldwide. So we all know this figure. I just want to remind you about this very important ecosystem service. Here we have a carbon burial rate in salt marshes, mangroves, and seagrasses. This is what we call coastal vegetated ecosystems. We could also add macroalgal beds here, but these are the three that have been studied more often, compared to terrestrial ecosystems and mostly forests. And so keep in mind, of course, the mangrove is a forest, so we can directly compare the mangrove with the terrestrial forests, but a salt marsh or a seagrass bed, these are not forests. So of course, the above ground biomass and also the below ground biomass in these two ecosystems is much smaller than in terrestrial forests and much smaller than in mangroves. But still, the annual carbon burial rate, burial in the sediment in these three coastal vegetated ecosystems is about by a factor 10 or even more higher than in most terrestrial, or essentially all terrestrial ecosystems. That means that even though these ecosystems, these coastal ecosystems, cover only a very minor proportion of the Earth's surface, they play a very significant role in carbon sequestration over time compared to terrestrial forests, for instance, that cover by numbers of magnitude larger areas in all over the world. And the interesting thing is, and you probably all have seen this figure too, the interesting thing is when we look at mangroves, at least in the Indo-Pacific, but now we know that similar pictures also can be found in other areas and other regions of the world. The interesting thing is when you look at the above ground biomass, the white part of these bars, this is pretty much the same in terrestrial forests and mangrove forests. There's not much of a difference. It gets a bit more different when you look into the upper centimeters of the sediment compared to the upper centimeters of the soils. There is already about twice as much per unit area, per hectare, twice as much organic carbon stored in this area. And part of this is the very dense root network, below ground root network of the mangroves. So mangroves have a very high below ground biomass relative to the above ground biomass, much higher than terrestrial trees. Maybe because of the stabilization issue in the sediment, maybe because the sediment is anoxic and they need more roots over a larger area to pop out into the air for gas exchange. We don't know exactly why that is, but we have quite some more carbon stored per unit area in mangroves than in terrestrial forests. But the largest difference, and this is really striking, is in the sediment below 20 centimeters depth. There's not much living roots anymore at that depth, but there is a lot of organic carbon, a lot of organic matter in general stored in these deeper sediment layers. And as you know, the reason for that is very simple. It is because these sediments are saline and anoxic. So organic matter under these saline and anoxic conditions is very stable over time, over decades, centuries, probably even millennia. Decay processes are very slow, and the material stored there is very stable. And this is the value of the mangroves and other coastal vegetated ecosystems as stores of organic carbon, as blue carbon in these ecosystems as climate change mitigator. An important point that I want to make here is just a side note, but you just saw the red line that I put here. We have to be very careful, and people are not always careful when they do that. We have to be very careful in terms of using different terms. So what we see here in this picture is not storage, but it's store. Storage is a process. That's the process of sequestering, of storing carbon in the sediment or in the biomass. But the outcome of that, what we see here, so carbon stocks per area, is a store. So please keep that in mind because when we talk, we should be accurate in order to be able to understand our partners that we talk to. The same is true for soils versus sediments, but I won't go into that here. That we'll need to find and will distract from the topic of this talk. So just some maths about the blue carbon numbers that we find in mangroves. And please, again, be aware, we have to be extremely careful with that. I'm very often asked by politicians, policymakers, or NGOs, or the media about numbers of carbon or organic matter stores in mangroves. And the problem is that these data are very, very variable regionally. And this is probably also a very important aspect when we talk about the Mangrove Health Index, which you are doing during this training. So we have different species that differ in terms of how much they store and how stable the organic material is. We have different environmental conditions. We have different sizes of the trees, so different biomass of trees depending on the environmental conditions. That means that there is nothing like a global average mangrove in terms of blue carbon or in terms of any other ecosystem services. Having said that, what we can still do is calculate some kind of an average, keeping in mind that it's not really representative for all mangroves worldwide. So when we do that, then on a global, very careful average, we see something that per hectare of mangrove, of an old grown mangrove, we have something like 900 tons of carbon stored in the biomass above ground and below ground and in the sediment. So this is the store. The storage, so carbon per year per hectare is about 2 tons in a mangrove. This is what a mangrove per year stores. And this is binding about the amount of CO2 that 4 cars produce per year. It doesn't sound very much, but then again, it's only 1 hectare of mangroves. And when we look globally, we have about 15 million hectares of mangroves. It's a tiny amount or tiny area compared to the tropical rainforest, but it's a very potent area. And the amount stored in the mangroves worldwide per year equals the amount of CO2 that is emitted by all cars in Germany. And that's, from my point of view, and I know how many cars we have on the German roads, I find this is quite impressive on a global scale. Then, of course, I don't get into that much. Of course, we have a heavy loss of mangrove area, but I have to say these numbers are not accurate anymore. Recent papers say that globally we are now down to less than 0.3% per year, which is good news. It's still too much, but it's very good news. And I won't go into mangrove conservation too much here. I just want to mention that we are losing a very valuable ecosystem in terms of ecosystem services to local societies and to humankind worldwide. So keep in mind, this is storage in the above ground biomass, the below ground biomass, and the sediment organic matter. And I want to show you a study that is not yet published that I'm involved in. That's an analysis of the Sundarbans in Bangladesh. And we did a study on above ground carbon, below ground carbon, and sediment organic carbon. And we wanted to understand, using structural equation models, we wanted to understand the contribution of different measures of biodiversity to these, how much they are drivers. And this leads back to the question that I mentioned in the beginning, that it is interesting to know which species are driving ecosystem processes that translate into ecosystem services. And we were very surprised to see that species richness, SR up here, has a relatively low explanatory power for either of them, above ground carbon, below ground carbon, and sediment carbon. Rather low explanatory value. Functional diversity, rather than species diversity or species richness, functional diversity was even less or even weaker in explaining the stores of organic carbon. In the end, it was not significant as a driver. What was highly significant is what we call functional distinctiveness. So it's not so much functional diversity, how many different species or how many different functional groups we have in an ecosystem, or in an ecosystem. Functional distinctiveness is more about how different are these species from each other in terms of their functional traits. So what we find out here, for above ground carbon, below ground carbon, and sediment carbon is that the major driver is how different the species we find in a given area are from each other. So the more different they are in terms of their functional traits, the more above ground, below ground, and sediment carbon is sequestered, is stored in the mangrove. So species identity plays a very important role here. I hope that we will be able to publish that soon, and you might be updated once you see it in the journals. So totally different area of the world, Brazil now, and you remember Atlantic East Pacific, much less species than in the Indo-West Pacific of the Sundarbans, but a very similar picture here. This is a paper that we are currently also trying to publish. It's a trait analysis, a global trait analysis of mangrove trees with a perspective of ecosystem ecology. So what we see here is underlined the four major, well, actually these three, Avicennia germinans, Laguncularia racemosa, and Rhizophora mangle, these are the three major species. We have another Avicennia species here, but it's not very common. So these underlined ones are from Brazil, from the Atlantic East Pacific. All the others are species of the Indo-West Pacific. And we did a coordination effort here with all the traits that we have had at hand. And this is kind of the landscape, the trait space for these different mangrove species. And the point I want to make here is that along these second axis, the coordinate 2, these three species of mangroves in Brazil are almost as different from each other as are all the other species in the Indo-West Pacific. On the first coordinate, I don't go into detail here, but on the first coordinate, the species are rather grouped by their genera. So this seems to be some phylogenetic or taxonomic traits, but along this coordinate, we have a very distinctive trait space by these Brazilian species compared to the Indo-West Pacific species. And why is that relevant? Again, remember what I told you before about functional distinctiveness in the Sundarbans. From my point of view, it means that this could be an explanation for ecosystem processes or the efficiency of ecosystem processes in Brazilian mangroves. When you look at the mangroves, at the photos that I showed you before, the biomass in this area of a mangrove is about the same, or at least not much lower than the biomass of mangroves that we find in many places in Indonesia or the Indo-West Pacific. And this would contradict the idea that biodiversity is a driver of productivity, because this hypothesis would predict that productivity, and in the end, then biomass of mangroves in Indonesia, would be much higher than in Brazil. But it's not. It's a bit higher, but not that much. And our explanation is that here, these species are different enough from each other to drive ecosystem processes as efficient as the many species in the Indo-West Pacific. So what that means, again, to kind of briefly summarize at that point, is that it is important which species we have in a given area when it comes to ecosystem processes, and then one step further, how that translates into ecosystem services. Which again means if we have to rehabilitate or restore a mangrove that is not healthy anymore, then we might want to think about which species we want to implement in order to make ecosystem services that are mostly needed being provided by the mangroves. So mangroves act species specific, and I just want to bring you one more example or another example, because one of the important contributors to the sediment organic carbon or sediment organic matter that is the important carbon store or blue carbon store in the mangroves is the input of organic matter from the mangroves themselves or from autochthonous material, seagrass, macroalgae, whatsoever from the sea entering the mangroves. And if you look at this, then you see the roots of Rhizophora back here, the prop roots, and the pneumatophores of Avicennia up here. And we can very well imagine that they differ in how efficiently they capture particulate organic matter, as you can see here in this picture. And yes, indeed, they did in a flume experiment with artificial root mimics. We tested how many leaves floating in the water are trapped by the different root structures. And it's interesting to see that when we look at fresh leaves that are floating on the surface of the water, then Rhizophora is much more efficient than Avicennia. And it makes sense because the roots of Rhizophora reach into the water and leaves floating on the surface will be trapped by the prop roots. When you think of Avicennia and the water level is higher than the roots, the pneumatophores are long, then the floating leaves, of course, will not be affected by the roots at all. When it comes to decaying leaves that start sinking down to the ground, then Avicennia and Rhizophora are about the same in efficiency, and they trap much more of the material. But again, we have species-specific differences between different genera, between different species of mangroves. And this is important because the organic matter that is trapped inside a mangrove depends on the mangroves, the amount, but also the origin. In an Avicennia forest, besides all the allochthonous material from the sea, we will also find a lot of autochthonous material from the forest. So the litter will differ in an Avicennia stand versus a Rhizophora stand versus a Bruguiera stand. And it's very clear, it has become more and more clear over the last 10 to 20 years that the origin, so the chemical composition of the organic matter drives its stability. This is what we call recalcitrance. So the residence time in a given system, in a soil or in a sediment, partly depends on the chemical structure, on the chemical composition of the organic matter. And that's all I want to show you here. I won't get into any chemical details here that will go beyond the aim of this presentation. Just keep that in mind, it's important. And I want to show you what we do with this in a case study from Colombia, from the Caribbean, Colombia. And for this, we wanted, so the aim from our point or our side is to understand the quality, the recalcitrance of organic matter of different origins in mangroves, in seagrass beds, in terrestrial forests, in macroalgal beds. And we developed a method, and again, I won't go into any detail. We developed a method that enables us to very clearly chemically distinguish mangrove leaves from seagrass and macroalgae. And with this, we found, for instance, more than 220 individual compounds that very clearly distinguish mangrove leaves from the organic matter of macroalgae and salt marshes. Only 20 of them are, 20 of them are specific for one species only. So we can, even on the species level, very clearly distinguish here between different sources of organic matter, keeping in mind that this is important in terms of recalcitrance of the organic matter, meaning in terms of the stability in the sediment. And this is what the blue carbon issue is all about. Blue carbon is only relevant or only valuable for climate change mitigation as long as it's stable, as long as it's stored in a stable form in the sediment. As soon as it gets decayed and mineralized, that will release CO2 or methane or N2O. So this is, from our point of view, an important step to understanding better what's going on in the mangroves or in the sediments. And I just want to show you the outcome, or one potential outcome of that, without explaining too much about it. So this is in the sediment, in a bay between a mangrove area and a reef out here. This is where we find mangrove-specific organic matter in the sediment between the mangrove and the reef, because part of the material is just outwelling from the mangroves into the open or into the coastal water, and other parts are inwelling. We expected more. We were hoping, well, we know we shouldn't hope as scientists, but we were expecting that the material from the mangroves would reach all the way out to the reefs, so we could show that the mangroves also fuel the reefs. But the reason that we don't observe that here is that all through the year, there's a very strong wind from northeastern direction, and that drives the water body into the bay, so the mangrove material cannot spread much. And at the same time, there's a longshore current that drives material all the way up here, because there's no mangroves around here in the area. The only mangroves we have is here and here, but still we found spots of mangrove organic matter up here. just details for the background information. We did the same for seagrass specific compounds. We did the same for macroalgal compounds. And here you see, interestingly, that inside the mangroves, there's a lot of macroalgal organic material stored. So with the wind and the wave, the macroalgae are drifted inside this bay of the mangroves and are stored in the sediment of the mangroves there. So the mangroves here are a trap, a sink of allogenous organic material, so also contributing a lot to the storage of blue carbon in this ecosystem, contributing a lot to climate change mitigation. So why are we doing all this? So very briefly, I won't talk about this project, but this is a project that we are running, or we are close to finishing it now, that we were running globally, 6 different sites all over the world, studying mangroves and studying the flora, the fauna, the microbiota, and trying to understand how the use of mangroves by humans, how the status of mangroves, or call it health, according to the Mangrove Health Index, how the health of the mangroves drives the community of mangroves and how this drives ecosystem processes. Let me explain in a bit more detail. So we are interested in the community of mangroves with all the different fauna species. And I showed you the different species, or the distinctiveness of species is important. The same for the flora, the same for the microbiota and the sediment, about which I did not talk today at all, but it's a very important component. So these 3 components act together, they interact, and they drive ecosystem processes. We talked about this a bit, at least. These ecosystem processes translate into ecosystem services that are made use of by humans, by local and regional societies, by humankind worldwide. So far, very straightforward. The important thing is now that the use of these services by the users feed back into the mangroves, into their species composition. If a certain species is cut, if a certain crab species is exploited or even overexploited. And also by this, it feeds back into the mangrove health, the mangrove status. And this, again, we can now imagine will feed back into the whole loop of ecosystem process, service, and users. And it's kind of a feedback loop that we have here. So we have to take into account, when we are interested in the ecosystem services, the use and the threat of the use on the mangrove health, on the species composition. And this will be important, this will prove important as a baseline for sustainable management, sustainable use management in mangroves. So again, why is that important, or what are we doing here? We are measuring community composition in the mangroves. So we can do that at present relatively easily. We can also do that going back into the past. I didn't tell you about that, but we use ancient environmental DNA to understand how the mangroves looked a couple decades, couple centuries, or maybe even a couple of millennia ago. We measure environmental conditions. We are, from the sediment, able to kind of back or hindcast environmental conditions hundreds or thousands of years ago, and we are able to model environmental conditions in the future. When we take into account that the services provided by the mangroves in the end depend on the community of the mangroves, so the species that are present, and on the health of the mangroves, then it is clear that these services are area specific, and the use of these species, these services, is also area specific when we look at a global comparison, and also specific for different groups of stakeholders. So in the end, what we want to know, what we want to be able to predict in relatively complex models from all this information is how mangroves of the future will look like, what these mangroves of the future will be able in terms of service provisioning, and of course also to understand what will be the future of the mangroves. I mean, they are threatened in many places, in other places they are used sustainably, but what is their future? For how much longer will they be able to provide all the services that we need, and particularly people living in and from the mangroves will need. So this is where my presentation, my lecture today, very directly links to the Mangrove Health Index that you are being trained during this week. So what do we make out of this? We developed a concept for spatial conservation prioritization that we call the traffic light concept. And in the end, something like the Mangrove Health Index is a very basic, very basis of this traffic light context. So we want to categorize mangroves and other coastal ecosystems into very simply into 3 categories. And we keep it simple in order to be able to communicate this with policymakers and decision makers, because they don't want complicated and complex concepts. They want it simple and straightforward. So we distinguish between the red traffic light, that means don't touch, protect strictly, because these are very valuable and they will remain valuable according to our predictive models, very valuable mangroves, but they are very delicate and very sensitive to disturbance. The yellow traffic light allows for some sustainable use. So these are still valuable and they will be valuable mangroves in the future, but they seem to be kind of resistant or at least resilient to stress or disturbance from the outside, including sustainable use. So here we would allow some use because we cannot protect people out of mangroves. There will be no acceptance in a given area, in a given region, when we tell people, don't use your mangroves anymore. They would not understand. They would not accept that. So we have to, if we say this region is protected, you cannot go in here, then there must be an alternative where they can go and where they can sustainably use the natural resources. The green traffic light is a go. It means we allow this area for more or less open use. This is very provocative, and we had a lot of controversial discussion about that, but I stick to it. It's more or less open to use because there the mangroves are lost already, or they are already so degraded that we predict they will be lost within the next couple of years. And that means, when I talk about open use, that means that we have to negotiate. If there is a company that needs to build industry or a harbor or a hotel resort or a golf course in any area, then they should not build it here where the mangroves or the ecosystem is either yellow or red, but they should build it just a couple kilometers away where the ecosystem is gone anyway. In a hypothetical space, that could look like this. This is the area, the Ajuretewa Peninsula that I talked about in Brazil before. This is a protected area. It's an extractive reserve, so people can go into the restricted area, preserved area, and extract sustainably some resources. And this is what we would have here. People can go for sustainable use. The mangroves are valuable. They will remain valuable. And they're relatively stable, relatively resistant. In this area, hypothetically speaking, we have the same value, but for some reason, this area is very sensitive to disturbance, so we have to totally close it, but still people can go here. This now would be the area very heavily influenced by the city of Bragança down here, so the Caete River, the estuary up here, and hypothetically speaking, we could assume that this area is very disturbed already. The mangroves are kind of degraded. It's not the case. Again, it's an hypothetical example. And there we would allow for use, for building a harbor, for whatever kind of use within given limits. But then, of course, there's an alternative to how we treat or what we do with a degraded area, an alternative to open use. And that would be kind of re-implementing functioning ecosystems. And this is what we sometimes do with ecosystem restoration. And I just want to introduce you to the concept of ecosystem design, which is also very closely related to mangrove health or to ecosystem health in general. But the difference, the very important difference between ecosystem design and ecosystem restoration is that restoration is aiming at rebuilding an ecosystem that is very close, very similar to what we had before. So restoration is mostly interested in ecosystem structure and biodiversity. That's often an aim. So it's more interested in the ecosystem itself. Ecosystem design takes much more into account the humans, so human needs for ecosystem services. Ecosystem design aims at re-implementing ecosystem services rather than the ecosystem, which means we need to know which species, as I had shown you before, which species are required to drive a given ecosystem service. And we've seen that different mangrove species and different crab species, different snail species, drive different ecosystem processes that translate into ecosystem services. So what we need and what we are currently developing is a toolbox, which is kind of a software, an app, if you want, if you wish, that gets or contains information about species and their traits. We talked a bit about that, and about the interactions of these species. We barely talked about that, very little only today. This together will give you will give you this toolbox of ecosystem design. And at the same time, a very initial step is that we talk and negotiate with local and regional stakeholders. What are their needs for ecosystem services? What do they really need from the ecosystem that we want to re-implement? And taking this and this together, the toolbox will tell us which are the species that we need to re-implement, or the species combinations that we need to re-implement in a given area, in a given degraded area, in order to implement ecosystem services in this area. So implement what humans need in this area rather than just ecosystem structure. And this is what we call ecosystem design. And with this, as I'm aware, quite controversial aspect, I'm done. And I would like to thank you for your time, for your attention, and I'm happy to get your questions. Thank you very much.
Thank you, Martin. This is very nice presentations, very nice lectures. I got a lot of information, knowledge, enlightenment, and of course, this lecture is complemented with the previous lectures in the morning about remote sensing, and then I thought you have already a lot of questions for discussions about ecosystem services and how we can use remote sensing for what we call it, for the designing the research that to answer the research questions that come up. Yeah, so please, for all the participants, just raise your hand and then ask questions, please. Yep, Jared.
Professor.
Please, Jared.
Sorry, go ahead.
Okay. Professor, I am Udara from Sri Lanka. Actually, I am studying also blue carbon dynamics in Sri Lanka. Actually, it's pretty much interesting field during these days when we consider about the global trends for the blue carbon dynamics. Actually, Professor, doing my research, actually I doing southern Sri Lanka, the blue carbon dynamics, not only mangroves, so tidal salt marshes, and also seagrass meadows. So during my studies, I think you focused on basically focused on the technical terms that you mentioned about the storage and store. Actually, I have a problem about 3 different technical terms. Which the first one is the carbon sequestration, second one is the carbon sinking, and the third one is the carbon storage. So could you please differentiate these 3 technical terms? Actually, it's very helpful for my studies.
Okay, thank you, Udara, for this question. It's a difficult and actually pretty tricky question. So I'm not very familiar with the term carbon sinking, and thinking about it, I might tend to try and avoid it because it sounds very unspecific to me. But maybe while I talk, maybe I rethink it and maybe I come to a different conclusion in the end. Let me just go to carbon storage and sequestration. So they are very, very similar. They're both processes. And I would say that carbon sequestration is very often considered more the build-in of CO2 into the biomass of the plant. Okay. This is at least my understanding. But as very often with these terms, you ask 3 experts and you get 5 different opinions. So we just have to be very careful, and we have to agree at some point. But this would be my understanding. Whereas storage is maybe more an overarching term that basically includes and maybe even focuses on the storage of organic matter in the sediment. So sequestration would more be the biotic process that the plants do. They sequester carbon in their biomass, and the storage would more be a also biological, but more physical process of bringing in organic material and burying this into the sediment. And maybe this would then also be referred to sinking, which I still think is a bit more vague, but maybe the sinking is the really the bringing in and the deposition of organic matter in the mangroves. But as I said before, I'm not familiar with the term sinking, so I might get that wrong. Because it could also, by word, it could also mean that this is kind of the burial and the kind of spatially entering into the sediment. The deeper you go into the sediment, the more the material sinks in. But I'm not sure this is the point here. So I'm sorry I cannot enlighten you about sinking, but maybe I made my point clear about storage versus sequestration.
So I have another question. So when we are finding above ground biomass, so there are specific allometric equations. So in my country, in southern Asia, so how could I access? How could I find some area specific allometric equation when finding above ground biomass? Because there are lack of allometric equation about.
You are touching a very delicate point, Udara. I mean, you know, and I guess most of you, all of you know, that people are using estimates of above ground biomass and even below ground biomass and sediment organic carbon, where this is a totally different story, but above ground biomass, they're using remote sensing data and trying to deduce above ground biomass from that. This is based on the successful attempt of measuring forest structure, tree density, tree height, diameter, breast height, all these things that you are aware of, using this to deduce biomass. This is well established, this is possible, but this is possible only, or it's accurate only, as you mentioned, if you use regional and species-specific models, allometric equations for that. And that's exactly the problem. Many people, many scientists, I would almost tend to say that they abuse these equations by not applying them properly, because they use an equation of whatever species or an average of different species from different regions, and that doesn't work. So, and this is exactly the problem that you mentioned. In many regions, we don't have these equations. And I'm not aware of, there is attempts to overcome this problem, but I'm not aware of any really suitable attempt, to be honest. I'm afraid the only way, and this of course is what we don't want, because it's a lot of work and we don't want to destroy the mangroves. But I think in the end, for a given region where we don't have any regional equations, we don't have any species-specific equations, the only way of getting accurate numbers might be to cut a couple trees, to dig out the roots and really measure their biomass after we measured the forest structure, the tree height, diameter, and all these things. I'm afraid to really get these numbers, this is the only reliable way. But of course, if you have equations from a nearby region, from closely related species, and you can make sure that the environmental conditions in your region or in your mangrove stand are similar to where other people develop these equations, then you can still kind of make an estimate, because the same genus, they might be different, or they will probably be very similar in their equations. And if the conditions are the same and the trees, if you don't, as long as you don't compare a stunted dwarf tree with a huge giant of a tree, then I think it still works in a given range. So bottom line of what I'm trying to say probably is, if you want an accurate model, then you need to do it. You need to develop it yourself. You need to cut trees and dig out roots. If you don't want to, or if you can't do that, then there is reasonable trust in equations from similar regions and similar species, but you have to be aware and you have to mention that, of course, you did not use equations developed specifically for your region and specifically your species. I'm not sure that answers your question, but this is my point of view here.
Yes, sir. Thank you very much, sir, for your kind answers.
Good, Dara. Next, Jared, please.
Thank you. Thank you for the great talk. I work in Singapore on plant functional traits, so I was very interested in your scatter plot of the different mangrove species based on their traits. So I wondered if you could talk a little more about what traits were significant and what the analysis was. I wasn't familiar with that. It looked like a principal component analysis, but
right. Yeah, thanks, Jared. It was a multidimensional scaling rather based on Mahalanobis distances, if you want any details here, and that's why we are not really able in this context to distinguish or to really assign specific traits to the different coordinates. With this kind of analysis, this is simply not possible. In other instances, we also did principal component analysis, and there, of course, we can assign different traits, but in this case, it was not. So we do not really know which traits these are that cluster around this coordinate where we have the high distinctiveness of the plants. This would be probably an additional future analysis that we would have to do. This analysis is totally based on published traits of mangroves. So some years ago, and we published this database, I think 3 years ago, we went through all the data available up to 2016 and just made a matrix of all mangrove species with I think it's 35 different traits. Most of them are quantitative. They refer to the leaf chemistry, to wood density, maximum tree height, stuff like that. And some of them are qualitative, like shape of seeds, root type, and all these things. So it's a mix of different traits that we're looking at. And the aim of the whole study, the whole analysis, was more understanding what makes mangroves different from other vegetation types. So, for instance, are mangroves more similar in terms of traits, more similar to wetland plants or to desert plants or terrestrial trees or whatsoever? And also to understand then what can we expect actually from different traits of mangroves in terms of ecosystem process and ecosystem functions. As I said, when it comes to this biodiversity versus productivity conundrum, then I cannot yet tell you which are the traits that we believe are important. Sorry for that.
Great, thank you.
Right. Good, Jared. Next questions from Isuru. Please, Isuru.
Hello, Professor Martin. Actually, I'm Isuru from Sri Lanka. Actually, I'm working on my master's, and my study basically look into how carbon is transported from one ecosystem to the next, the ecosystem. So basically, what you said about this allogenous carbon. So I thought you might be the best person to ask these questions. So I have like a couple of questions to ask from you. So my first question would be, so in your presentation, you mentioned about the flume technique. So what I'm going to also incorporate in my study is a flume flow-through system. So could you please explain me about what sort of factors that we want to focus on when setting up a flume flow-through system? That would be my first question. And the next question that I have is, so I have searched a lot of literature about these flux studies. So And most of the studies that I searched were basically all those flux calculations, the equations have incorporated this tidal variation, the flood and the ebb tide when calculating the fluxes. So I'm having this problem, especially like in my country, we have a microtidal, like very minimum tidal inundation, tidal variation. So in my case, I have to apply this flume technique to mostly to unidirectional flows. So how could we use, how could we amend this flume technique, the flux calculations to unidirectional flows as compared to the bidirectional tidal variation flows? So that would be my second question. And when it comes to the third question, so in flume techniques, we basically calculate the flux of carbon, which is through water medium, right? So I'm also focusing like how could we do some flux analysis which happen through other sort of vectors, for example, maybe leaf litter, maybe sediments, and even through active vectors such as fish and other species. So my question is like, what sort of techniques that we could basically use to calculate fluxes of carbon, which other than the water? So those are my questions. It's quite lengthy, but I really appreciate if you can provide me with some advices.
Yeah. Thank you, Cyril.
Yeah.
Indeed, connectivity, or I usually call it connectedness of these coastal ecosystems, is hugely important for the whole ecology of the system, but particularly for the blue carbon. So I totally agree. So when it comes to the flume, how to design the flume or the factors that you should take into account, that of course in the end it depends on the flume that you have available. But what most people do is, most of the studies that I'm aware of, not all, but most that I'm aware of, are kind of actually work with a unidirectional flow. So they have this water flow running through their either real planted or mimic mangroves or seagrass bed or saltmarsh plants, and then they study processes of sedimentation or particulate organic matter deposition. And that's very helpful. And this is also what we did with the one paper that I presented. It's very helpful, but it's only a little part of reality, of course, as we all know. So bidirectional fluxes is very important, first of all, because if you don't, even in a microtidal system, you still have that. You still have very little, but you have some incoming and outgoing tide. And this is something that you really should model in such a flume. I would say that you really take into account there's a time of incoming water, so unidirectional flow at one point, and then unidirectional flow in the opposite direction at another point. That's still relatively easy, again, depending on the flume system. It's getting much more complicated, and there we have a very strong bidirectional aspect in the water movement. It becomes very complicated when you want to take into account wave movement. And this is a super important aspect in spatial dynamics of the water body in the mangroves, because it makes a difference whether the tides come in just as a water body very slowly and very evenly into the mangrove and then flow out again, because in the end, at a given time, it's only unidirectional. With this, I agree with you. But then at the same time, we have, or it makes a difference if then you have waves on top, and in many cases we do have waves on top of this tidal elevation change or tidal height change. And these waves make it very complicated, maybe not complicated, but complex, because this is bidirectional. The waves come in, they hit the mangroves, they are slowed down, they're attenuated and all these things, but still the water mass that came in with the wave will go out very shortly after it entered. And that's the bidirectionality within a very given time, a very short time. And if you ask me, from my point of view, this is something that really needs to be studied in terms of fluxes of particulate organic matter, but also dissolved organic matter, and of course sediment. So if your flume, or if you have access to a flume system that can do that, my recommendation would be to focus on that, because this, I have to think, nothing pops up immediately. So I think the number of studies that did that are close to zero. And I think there's a good reason for that, because it's technically challenging to do it. But I think that would be a very interesting point to take into account, this back and forth of waves on top of the tidal change that is an inflow at one time and an outflow at the other time. That was the second question. And the third question was, was about the unidirectionality and the bidirectionality. I'm not 100% sure whether I understood, because in the end, as I said, the tidal water movement, even if it's microtidal, is over time still bidirectional. So for me, it's just different time frames, whether you look at the unidirectional inflow and the unidirectional outflow of the tides, or the bidirectional in and outflow of the waves. But maybe I didn't quite get your question. Maybe this is not the answer to your question.
Actually, that's what I meant. Like, yeah. So maybe mostly the places that I'm going to look into is like mostly like rivers, like river dominated setups. So that's the question that I had. How could we apply this flume system? Yeah.
That is even an additional level of complexity that I didn't yet mention here. If you don't look at the coastal mangroves, but at an estuarine mangrove with a river discharge, well, that's wow, that's extremely challenging for a flume experiment, because you have well, it's not constant, but in a given time frame, it is relatively constant, the outflow, the discharge of the river. And then you have the tides. Well, then you don't have the tides in Sri Lanka because you have a microtidal system. And this tidal water movement is probably negligible compared to the river discharge. So you are lucky then, because that makes it easier for you. Because if you just have the waves acting against the river discharge, but in the end, yes, then in the end the water movement is more unidirectional from the river towards the sea. But if you wanted to take tides into account, then you have this changing force of the incoming water that counteracts the discharge of the river. And then at some point after 6 hours, it all reverses, and the outgoing tide even increases the river discharge. Wow, this is crazy. Modeling that or mimicking that in a flume, that would require quite some effort of construction, I would guess. So lucky you have only unidirectional systems.
I'm just wondering, sorry for taking time, but I'm just wondering what sort of materials that we basically use in flume systems, like does the material really matters when it comes to flume through system?
I think in the end it does matter, because the physical characteristics of the material. I mean, you can, when you want to mimic a pneumatophores of Avicennia, of course you can use little bamboo sticks or wooden sticks or even metal sticks, but they're not the same as because a pneumatophore can bend in the water. And we did some fine scale hydrodynamic modeling that this bending of a pneumatophore, or even the bending of a thin Rhizophora root in the water, that has a strong influence on the hydrodynamics, whether it's a stiff and rigid structure or whether it can bend with the water movement. So my advice would be, try and find something that in terms of physical characteristics is as close as possible to your root structure that you want to mimic. Do not just use a wooden stick, but try to find something better than that. If you can't, then the wooden stick would give you a first idea, but
good luck, Isuru, for your research.
Maybe
thank you so much.
Else, but so if you want, we can discuss by email. You had the question about
that would be really great, because I have loads of questions to ask.
No, please feel free to send me an email, and whenever I have time, I will answer. Okay.
I'll get the email from maybe.
Sure.
Yeah, I will share.
Thank you so much.
Okay, next question from Muhammad Soimin. Please, Muhammad.
Okay, hi Martin. What lecture anyway. I'm Indonesian. I have one question actually. In my island where I live, there is an assemblage of mangroves in which a couple of years ago it was dead suddenly. And I'm wondering what's the cause of that mortality, but I'm also wondering what approach of such a methodology that I can use to seek for the answer for that. I think that's it.
Thank you, Muhammad, for the question. It's that's a difficult one to answer from here in my home office in Germany. But of course, you need, I mean, there's many potential reasons why you might have a relatively large scale die off of a whole mangrove stand. Spontaneously, I would think of something like a chemical, I don't know, eutrophication or pollution whatsoever. It could be climatic. We know from Northern Australia that there are large mangrove patches that in a long-term cycle, every, I don't know how many decades, just die off because of climatic events. But I'm not sure this applies here. You wanted to say something?
Yeah. Actually, those mangroves are, I mean, like the mangroves which were that is actually in between of the others live mangroves. So
okay.
What's the explanation of this actually?
I'm afraid from here there's not much I can
okay.
tell. This is something that really needs to be studied in the field. I would look at, is there any reason why the hydrodynamics might have changed? Is there any potential influx from the hinterland of agricultural runoff? It kind of is channeled into this stretch of mangroves, but does not touch the ones next to it. Any currents that hit at this mangrove that might bring pollutants or whatever from the sea side. This is really, you would really have to go and check what is the difference between this one, I mean, environmentally, between this one stand and the mangroves next to it. If once you are able to answer that question, then this is the essential step in before you go for any rehabilitation, because you have to make sure that the factor that caused the die-off of the mangroves is gone, that you remove it. Otherwise, you can invest as much time and effort as you want, it will not work. So it's a very vague answer, but that's all I can, a very theoretical answer, but that's all I can do from here.
Okay, thank you.
Right. Next questions. Hangar, please.
Hello, can you hear me?
Yes.
Okay. Yeah, thank you very much, Prof. Martin. Is that I love for your presentation, and then it's my open mind for me. And then I just wondering about maybe talk about the mangrove ecosystem in here. We have case in Indonesia also is we call it is mudflat, and then me now is still studying about the mudflat, and could you tell me about or you have any opinion about what is the mudflat, and can that the mudflat can identify in the mangrove area with the remote sensing or technical in field, and just wondering about the research about the carbon in the mudflat in the world. Yeah, thank you.
Yeah. Thank you, Hangar. So this is this mudflat is a mudflat in front or next to a mangrove, or it's an open space inside the mangrove?
Open space inside mangrove.
Okay, how large is it?
I don't know. In my case in Bali, it have like in 3 hectare area.
Okay. Wow. Okay. Okay. Yeah. Again, that's a different one, a difficult one. It's so there's, if it's a mudflat, I assume there's no standing dead trees or no fallen dead trees on the ground. There's just an open area where there's no vegetation, a salt flat, a mudflat whatsoever. So this, we know this kind of open spaces from salt marshes quite well, actually much better than for mangroves. And there the reason is that there is some kind of either there's an elevation, a small scale elevation that increases, I mean, that changes the inundation scheme or dynamics or the water table below that would then affect the vegetation. But that's again difficult to tell without being there and without doing any studies. Another opportunity or another potential reason is that there is some hydrodynamic forcing or some reason, a barrier to the tidal movement. We have this, I know a case pretty well from Brazil where there was a road construction, and I assume in your case that is not the case, but there was a road construction that kind of cut off a huge mangrove area from the tidal flux. So the tides still came in, but then the water could not go out because of the road. So the water stayed in place and slowly evaporated. And that increased sediment salinity by factor 5 or something. So when we go there now, we measure sediment salinity and we get values of 90 or even above 100. And this is a kind of a bare area, or it has been a bare area for decades. Because most of the plants that we know are not capable of standing these conditions. At some point in this area in Brazil, Avicennia started to recolonize, and they somehow managed, but the trees are like that high, so they're really, really tiny little dwarfs, but they kind of still managed to survive. So I don't know whether this might be the case in this area, whether there's some physical hindrance from tidal water flowing out again after it came in, and you would have a very high salinity in that mudflat. This I don't know. This is something you would have to measure. You would have to check by inundation loggers or by observations in space. But again, as before, the question by Izuru, it's very difficult to answer that from without going there and having a look at the place. You could also analyze the sediment chemistry and check what is in the sediment or what might be the difference between the sediment there in your mudflats inside the mangroves and inside the mangroves themselves. That could also explain at least why the mangroves don't grow there, but then I would still expect some other, like for instance, salt marsh plants to get there. Do you know how old this gap is, this mudflat? Has it been there forever or is it recent?
I don't know. I don't know how old that for that. Yeah, just wondering because maybe it's 5 year, and now when we we have accident that when we planting the mangrove on there, so every gone is every die, and why that is need answer for that. Yeah.
Then as I said, then I would go and analyze the sediment for various characteristics, grain size, chemical composition, maybe even have a look at the organic matter there. And at the same time, I would try and figure out the dynamics of the tidal water movement. What is going on during a tidal cycle? And this might help answer the question why there is this mudflat. And this answer would then also be the answer to the question why restoration efforts don't work. Because there's a reason that the mangroves are not there, and if you don't change anything, then mangroves will not establish at that place.
Yeah.
Thank you.
Wow. That's enough, Hanggar. I think it's fruitful discussions.
Yeah, maybe I if you mind, I will discuss you with email also with Mas Yahya.
Sure, absolutely.
Okay,
Thank you.
All right. Next questions. Gary, are you there? Gary? Please, Gary, for your question.
Yeah. Hello. Yeah.
Yep.
Hello. Okay. Thank you so much. Hello, Martin. Thank you so much for the very interesting presentation. I would like to ask of your insights about the role of the mangroves in terms of deposition, because you have discussed also about the berms, about in the coastal formation. Because we have recently we have this study and we found out that the mangroves, the fringing mangroves, has this effect, which is actually more on negative side because it actually embeds the deposition of the sediments that leads to the lesser formation of the coastline in certain area. What can you say about this one?
That's very interesting. Thank you, Gary. It's very interesting because it I wanted to show you during the presentation, I wanted to show you the outcome of some hydrodynamic modeling that we did. I just mentioned it a couple of minutes ago. I decided not to present to you because it would have taken too much time. But this, what you just said, this kind of confirms some modeling outcomes that we had recently, where we saw that around, well, we modeled a structure that would be similar to a Rhizophorus seed that just got stuck in the sediment, or a pneumatophore of an Avicennia or Sonoraceae, so very similar structured. We modeled that, hydrodynamics and sediment dynamics around that, and we found a very heavy effect of scoring. So around these structures, sediment actually is not accumulated, but is lost from this area because of turbulence in the water. So this is something that in the beginning we thought this is not possible. There must be a mistake in our model. So we went back again, we tried again, the outcome was always the same. And then we found actually evidence that this also happens, that this can be observed in the field. If you go to mangrove, a mangrove stand of Avicennia Sonoraceae, then you will see little horseshoe scores around these pneumatophores. And even on a larger scale, we saw satellite images of individual mangroves in a very peculiar area that show these scoring around their stems, around their root system. So obviously, in some instances, mangroves do not accumulate sediment or deposit sediment around their structure, but the dynamics, the turbulence of the water body even removes sediment, which is kind of counterintuitive when we think about coastal protection and sediment stabilization. So our interpretation is that in the end, and we are currently trying to test this again with models, one hypothesis is that maybe the spatial density, how many structures do you find per meter square, and how are they arranged relative to each other? Do they occur in a row, or do they occur randomly, or whatsoever? That this might have an effect on whether we have a net deposition or a net removal of sediment around the structure. Our models also showed us that, of course, part of the sediment that was removed around these structures has been deposited then, I don't know, 50 centimeters away somewhere. So it's not lost from the system, but it is moved around. And what we know by now from these additional models is that if structures are in a row along the water current, then they have this has an increasing effect on the sediment removal. Whereas when they are spaced kind of randomly, this effect is not improved or not strengthened by the addition of structures, which then, just as a side note, would have very strong implications for restoration efforts, because it would tell us that we should not plant seedlings along a row, but we should rather plant them some kind of randomly. So your findings are very interesting because they kind of confirm our model. I don't yet fully know the answer why that is, but I think this is something we have to take into account when we talk about sediment dynamics in mangroves. Thank you very much for this hint.
Thank you.
Good. Then we have Udara again. He still has questions. Please, Udara.
Professor, I want to know about the factors that affected to the carbon sequestration or storage or in the mangrove ecosystems. Is there, spatial or temporal? You mentioned that you are doing research on 6 different regions worldwide. So I think I'm just wondering to ask that, is there any, what are the factors that affected to carbon sequestration?
There is, thank you, Udara. There is huge regional influence on the blue carbon storage and stores in the sediments. And one very important aspect certainly is the tidal regime. And this is nothing new. I mean, this has been around in the literature for a while now. We know that in a microtidal system, there is much less outflow of organic matter from the mangroves into the open water or into the coastal water. And of course, much less inflow from organic matter from the sea, which first of all means that the that the organic matter that we find in a mangrove, sorry, with a microtidal system is mostly autochthonous. It mostly stems from the mangrove itself. And at the same time, there is very little outflow of organic matter from this mangrove into the water. So this question that has been in the literature, are mangroves sinks or sources of organic matter, totally depends on that. So this is one important point. We had this, I showed you this picture from San Andres, this island in Colombia. This forest is an inland mangrove. It is inundated once a year after heavy storms in fall. And then the water stays inside for, let's say, 3 months, maybe 4 months. It slowly evaporates. And then for the rest of the year, the mangroves stay kind of dry. There is no chance whatsoever that any organic material, any autochthonous material from the mangrove is washed out. And I think this is the reason why these mangroves are so luscious, because there's huge, and actually the amount of organic matter in the sediment is about 10 times higher than in many other mangroves that we measured before. So there's huge regional variation. And this is one factor that affects it. The species composition, the density of the trees, of course, has a huge influence. And this is one thing that I really want to, that's why I'm grateful for this question, Udara. It's really important for me to mention that there is no global average mangrove with regard to no characteristic, and certainly not with regard to blue carbon. There's always site-specific differences that we have to take into account when we talk about blue carbon. Unfortunately, this is not always the case. People talk about, we develop we develop global maps of organic carbon or organic storage in the mangroves that are based on extrapolations that are just not valid. So I'm not sure I answer your question. If not, just ask again, but I think this is my point of view on that.
No, so actually it's the answer because I'm just wondering that how could the carbon storage and sinking is differ from the region to region, and also those kind of factors. So thank you very much, sir.
Good.
Good. Professor Martin, I just share your email to the participants here in the chat room. So everybody, if you still want to discuss with him, you can just email. Yeah. And for the last,
your emails are very welcome. Just be aware that it might take me some time to answer, but I will answer all your emails.
Yeah. Okay, we still have time. I give the opportunity for Hung or Hung, sorry, for the
Hung.
Hung, yeah.
Yes.
Yeah, for Hung. Yeah, that will be the last person.
That's the last. Okay.
Yeah.
Sorry.
Right.
Okay. Thank you very much for Professor. I also would like to have the question about your concept. You give the three concept with the traffic light, right? And also to protect tricky and sustainable yield area, and also open yield area. So I would like to add what's the indicator for identify the each area that in the field.
Right. Thank you, Hung, for this question. It's a really good one. So the short answer is, in the end, the indicator that we want to use, and we're just in the phase of implementing that. We are just in the phase of talking to colleagues and policymakers in different regions in the world to develop this concept. We haven't yet implemented it. But the idea is that in the end, short answer, what we need for such a system is a mangrove health index. This is exactly what we need for being able to implement our traffic light concept, because we need something that describes the status and the stability, if you wish, of the system. So the status will be based, and you will learn, or you did not know, I think you will learn about this a lot during this training. The status will include, of course, species composition, and not only of the plants, but also of the fauna, and ideally the microbiota, and the characteristics of the vegetation that will tell you a lot about the status. And that will also tell you a lot about the potential stability and resistance against external disturbance. But we also need to understand, of course, the environment, environmental conditions, how they changed over the last years, and then, and this is the tricky part, then also the prediction, how will they change over the next 10, 50 years? And there we, of course, we are aware of that. There we come into something that is potentially a bit vague, because we have models, but these models are on a very small scale. These models are sometimes still not really working properly. So what we need to know in the end is, how did our system come to the state that it is now, under which conditions, under which pressures, under which drivers, and then take the current situation, the current pressures and drivers, and forecast how will this change the system in future. When you look at climate change, how will climate change, how will warming change the species composition? Because, and then species, when you look at species distribution, then you also have to take into account interactions of species. So if you have a different community in this place, how, or if a given community, how will this community change in the future? Sea level rise, how will the different species in this area respond to sea level rise? So that means you first need to be able to predict sea level rise in this area, which is very region specific. There's no global sea level rise. It's very regionally specific. You need to know that. You need to know how different species respond to sea level rise, whether they can expand landwards, whether there's space for expanding landwards. So the answer, besides the very simple answer that we need a mangrove health index, the much more complex answer is that it's super difficult to predict that. And this is why this traffic light concept, I think, still needs a lot of work before we can really use it as what we want to use it. But that's kind of the work we're currently doing. Does it answer the question?
Yeah, that's right. Yeah,
okay.
And because you know, we are the scientists, so difficult to explain for the government to know what we should keep this mangrove area, why we should protect, and also they also ask why and what indicator you mentioned, and you need to indicate that this mangrove area should protect tricky or can open you. Yeah, so that's why I would like to ask you about that.
Right. Yeah, well, then you're in a lucky situation that your government asked you that. I think in many places they don't want to know the whys and hows, they just know your recommendation.
Yeah.
So yeah, but I think it's very complex, and you're right. The challenge is, there's a twofold challenge. One is the scientific challenge. We need to understand, and we need to be able to predict. But the second one is, we need to be able or learn how to communicate with politicians and policymakers. What we are doing is really complex. I mean, we are still learning. We've been learning over the last, I don't know how many decades, and we're still learning. It's very complex, but it's way too complex to explain this to a policymaker, to a decision maker. We need to simplify and to explain in very simple words. And this is why we still believe that our 3 different categories of the traffic lights are very helpful for policymakers if they are reliable.
Yeah. Okay. Thank you.
Okay. Thank you. We still have 6 more minutes. If there is any questions, simple questions, only one simple questions. If there is, if not, maybe I got a simple question for you, Martin. You said about ecosystems design. Do you have any example in the world maybe you have already done with this concept?
No, we don't have an example yet in terms of that I can show you any successful case. It's the same as for the traffic light. We are just in the phase of implementing. Okay. But we are, and actually this is something that applies to Indonesia. We will start a project next spring in Kalimantan on what we call integrated mangrove aquaculture. And we are applying for a big project that will run over, I don't know, in the end, I think it will be 7 or 8 years. That is will also take place if we get the funding with colleagues from Indonesia, then spread all over the country, not only in Kalimantan. But this is an example that maybe I can talk a bit about, because the idea there is that we really find out Well, the ecosystem service that we need, of course, is the production of shrimp or whatever is produced in the aquaculture. But at the same time, we want to get some of the other benefits of mangroves back, be it coastal protection, be it carbon storage, or maybe even that a partial cover of the ponds by mangroves even increases the productivity of the ponds, which we don't know, but it might be. So this is the basic idea. So and then we would go and see, okay, if this is the aim, what are the species that we would need for? Which tree species do we need an additional crab species, or maybe anything else that we don't know yet? So it's still very theoretical, but we are now after 3 years of developing this concept, we are now in the first phase of implementing it. And another, if I still have a minute, another example is we got a request from actually a hotel resort from a very small Caribbean island. So this hotel resort owns a bay that in former times was inhabited by dense mangroves, but because of construction in front of the bay whatsoever, this changed, and now they're asking, can we restore mangroves there? And we started discussing with them, yes, of course. Well, maybe not. We have to go and have a look. Maybe the situation changed, so we cannot restore them. But in the end, what do you want from them? And in their case, it's tourism. They want people. They want to attract people to their resort because they will have beautiful mangroves. And there, the aim would be beautiful, diverse mangroves. So there, the concept is very simple. We would go for implementing as many different mangrove species along the tidal range as possible, and then maybe even trying to push the crab fauna by implementing. They will come by themselves anyway, but that will take time. So we could even push this a bit further. So this is. I hope you can get the basic idea that is behind it. It's just a further development of rehabilitation with a different focus.
Yeah, it's very interesting, Martin. Please, for you, maybe you have a closing remarks for this lecture and discussions is very good. So please, for giving closing remark for you, and then after that, I will close this lecture.
Sorry, I didn't get that. There was a.
I mean, do you want to give a closing remarks or closing statement? Yeah.
Well, yeah, maybe the closing remark would very simply be, and this is why I'm really very pleased and honored, Yaya, that you invited me to give this lecture in the context of this training, because really, this is something that has not been taken into account often enough. And whenever you do mangrove research, I would just pledge that you go in this direction, that we have to take into account this species specificity of mangroves and of the processes. A mangrove is not a mangrove is not a mangrove. They differ regionally, even locally, very much, depending on the conditions, depending on the species that we find there. And this is something that we need to take into account in mangrove research much more than we did so far. So maybe this is the take-home message.
All right, thank you, Professor Martin. We are closing to the end. And then, again, thank you very much for Professor Martin and for the fruitful discussions in this afternoon, in the morning for you, Professor. And yeah, this is very good for us. Many knowledge, many enlightenment, and then that's very good. But we, before we go, we need to take a group picture. Mbak Amel, please.
Okay.
Please.
Please open your video.
Yeah, show your face, please. Yeah, for all participants. Shaheen. Yeah, thank you.
Okay.
Buchanov, could you please to show your face?
Okay.
Not yet. Yeah.
Yeah.
He's coming.
Start
she's coming.
from page 1.
Yeah.
1, 2, 3. Cheers. Wait for a minute.
Yeah. No.
And then next, the last page. 1, 2, 3. Cheers.
All right.
Okay.
Thank you,
Thank
Amel.
you.
Yeah. Thank you, Professor Martin.
Thank you
Yeah.
very much, Yaya, for the invitation, and thanks to everybody for this very interesting discussion and for your attention. Thank you very much, and enjoy this training. You will benefit a lot from it.
Yeah.
Thank you, Professor.
Thank you so much.
Thank you, Professor.
Thank you,
Thank
Professor.
you, Pak Yaya.
Thank you,
Thank you for all
Thank you all.
participants.
Yeah.
Pak
See you
Yaya, Bu Amel.
tomorrow. See you tomorrow.
Yeah. Thank you. See you tomorrow.
Mas Yaya, langsung mati.
Ya. Langsung mati. Enggak, mau ngobrol dulu. Nanti tak bilangin ke Mbak Mamel jangan ditutup dulu kalau mau ngobrol.
Mau ngobrol.
Apa Mas Anggar? Seru ya Mas Anggar ya?
Apa? Iyo.
Seru.
Ih, Bapak ini keren sekali. Mas Maya nemuin dari mana ini?
Sorry Home, we are talking